Hi, I'm Jacob Plant, and I'm here to talk to you about phages. I know this thing looks a little weird, so we're going to go through this together. Now, I think it's safe to assume that most of you have at least heard of antibiotics, even if you've never taken them. But what you may be less aware of is that there's a risk posed by bacteria that become resistant to these medicines. And these antibiotic-resistant pathogens are a very real and dangerous public health risk, as they can be harder to treat and have higher mortalities than their non-resistant counterparts. In 2019, the CDC estimated there were 2.8 million cases of resistant infections in the US, resulting in 35,000 deaths. Since it can take more than a decade and billions of dollars to develop new antibiotics, where resistance can arise spontaneously, it would seem prudent to develop alternative ways to combat these pathogens. Enter phage therapy, which uses bacteriophages or viruses that target bacteria to treat bacterial infections. In order to explain the merits of this treatment, I want to share with you the effects of phage therapy on its own, as well as how it compares to more normal antibiotic treatment. Then I want to talk about the downside risks of using phages medically and other potential curatives for the antibiotic resistance crisis. By the end, I hope to have convinced you of the benefits of development of phages as a medical tool. Though people had been using antibiotic substances before, the first true antibiotic, penicillin, was identified in 1928, and then had entered widespread use in medicine by the 1940s. New antibiotics were still discovered periodically, but the newest class of antibiotics was developed in 1987, and there hasn't been any major development since. And some antibiotics remain widely viable for decades, but others can still become resistant in less than a year. So the use of phage therapy would require a separate form of resistance that bacteria don't easily accommodate alongside antibiotic resistance. This would allow for new effective treatments that can be safer and more effective than antibiotics. On an introductory level, phages are fairly simple in terms of infections. They attach to a host cell and inject the genetic material. And then this genetic material is used to make copies of the phages, as well as several enzymes. And these enzymes act as a sort of clock in this whole process, because once enough have accumulated inside the cell, they begin to create holes in the cell wall and cell membrane. And this will allow the newly formed phages and the cell's other contents to leak out, killing it. And this whole process is called the lytic cycle. So you may be wondering, what happens if these phages infect me? And the long and short answers are, they won't. Phages are very specific in their host choices, and many will only infect a single species or even strain of bacteria. And though they are similar to the viruses that infect humans, they can't infect our cells themselves. In case you wanted proof, phages are part of a happy, healthy human microbiome, just like the bacteria they might infect. This is the basis of phage therapy, administering a phage cocktail to a person or at the site of infection, and allowing the phages to clear out the offending pathogen without harming the patient. Fortunately, other people have already done the hard work of compiling reports on phage therapy. Liu et al. Found that the effectiveness of the treatment depended on how sick the individuals were and how the phages were administered. Phages passed through patients who weren't infected when administered orally, but injecting them almost always generated an immune response, which resulted in the phages being neutralized. Overall, they determined that in human subjects, the phages were well tolerated and most patients improved by the end of treatment if they didn't fully recover. In terms of how phage therapy compares to traditional antibiotics, Lin et al. Found several notable differences. Phages tend to have more subdued patient reactions and were able to operate where antibiotics failed. Of note is how phages were able to treat biofilms, which are made of bacteria surrounded by a kind of slime that prevents immune cells and antibiotics from attacking the infection. However, antibiotics were much less costly to produce and administer, both in terms of time and money resources. Aside from the potential costs, which will depend on developments in technique, Luke Carrillo and Abaddon found that not all phages are viable for use in treatment in the first place. For a phage to be considered, it must only use the lytic cycle to reproduce, that is to say, it must reproduce by killing its host cell. It must be able to be stored under reasonable conditions, and it shouldn't produce toxins during the process, among other criteria. Contributing to this is also a potential benefit, the specificity of phages, as a mistake could result in a phage that doesn't infect the desired pathogen. Of course, the immune responses triggered by phages themselves must also be monitored, as a severe response could make a patient's condition worse and lead to their death in severe cases. While some of these issues are not particularly difficult to overcome, others, like the potential costs, are troublesome. With this in mind, alternative methods to handle antibiotic resistance might seem more appealing. And Ventola found that the biggest contributor to the current situation is overuse of antibiotics in various forms. It's proposed that more strict requirements for the prescription of antibiotics for non-serious infections, as well as reducing usage overall, especially in agriculture, could be a remedy. Additionally, reducing barriers to the development of new antibiotics, including the creation of government grants and a review of regulations in place that moderate development, could also create new opportunities for antibiotic development. A quick recap before we move on. So far, we've seen that phage therapy works to treat bacterial infections, and it's possibly more applicable than traditional antibiotic treatments. However, it's more costly, and the downsides associated with it are mostly logistic in nature. There are also other ways to fix the antibiotic resistance crisis, and most of these involve reversing course on current practices that are contributing. Now, there can be other benefits from learning about how phages would work aside from wholesale phage therapy. As we've seen, the triggered immune response can have a serious negative impact on a patient's health, and engineering phages on a large scale could prove more challenging than we anticipate. And so a potentially more effective alternative is to use lysins to kill cells. Now, lysins are one of the enzymes produced that actually break open the cell wall, which I'll remind you is what actually kills the cell, which is our goal here. And so what we can do is we can create a lysin cocktail instead of a phage cocktail and simply inject that at the reaction site. And that would work much like the latter half of the lytic cycle, where they would implant into the cell wall and then break it open, releasing the cell's contents. Although this time, there are no phages to be released as well, which means this system is self-limiting. Some of the other reasons that lysins may be better include their smaller size compared to phages, which will make them less likely to trigger an immune response, the fact that it's easier to engineer a single enzyme compared to an entire phage, and last, it might be more difficult for bacteria to develop resistance to this form of treatment. Ideally, we would modify lysin enzymes with a domain that could attach to the outside of a cell wall as opposed to the inside, and it would begin breaking it down that way. Now, this reduces the risk of a resistance being developed because it's not actually interacting with the cell other than breaking it down. So there's no chance of some sort of internal mechanism breaking down the enzyme before it attaches and does its job. Now, even if phage therapy is determined to be the best course of research, it would still help in more than one way to counteract antibiotic resistance. The first would be the aforementioned fact that the phages would necessitate a different kind of resistance compared to antibiotics. And second, phage resistance is more genetically complex and resource intensive than antibiotic resistance. So it isn't an even trade-off for the bacteria to switch between the two. This means that a combination of antibiotics and phages would have a higher efficacy than either would on their own, because to resist the phage, it would have to lose its resistance to the antibiotics and vice versa. So what's left before we can start using any of this in medicine? Well, we need more controlled trials with humans to better understand how our bodies might react. Coincidentally, phages identified in humans don't seem to be much like what we find outside of our own microbiome, and the specific factors that we need to better understand include how dosages and toxicity are determined, i.e., what are the parameters we need to know for safe usage, how the cellular debris released will affect the surrounding tissue, and how to reliably and affordably purify the cocktails of contaminants. I think that all of this will be easily doable once we direct resources to the development. Most of the roadblocks we discussed here would have to be faced by basically all modern medicines as a standard part of regulations. While I myself think that the use of lysins will be more practical than whole phages, to do either, we will need to get a better understanding of how the phages themselves work in this medical context. I believe I've shown that the foundation for phage therapy is ready, and that has real potential as a solution to the threat posed by antibiotic resistance.